Phytoplankton Phytoplankton are the autotrophic components of the plankton community and a key part of oceans, seas and freshwater basin ecosystems. The name comes from the Greek words 𝝿 nu, meaning plant, and pi lambda alpha gamma kappa tau omicron sigma, meaning wanderer or drifter. Most phytoplankton are too small to be individually seen with the unaided eye. However, when present in high enough numbers, some varieties may be noticeable as colored patches on the water surface due to the presence of chlorophyll within the cells and accessory pigments in some species. Ecology. Phytoplankton are photosynthesizing microscopic organisms that inhabit the upper sunlit layer of almost all oceans and bodies of fresh water. They are agents for primary production, the creation of organic compounds from carbon dioxide dissolved in the water, a process that sustains the aquatic food web. Phytoplankton obtain energy through the process of photosynthesis and must therefore live in the well-lit surface layer of an ocean, sea, lake, or other body of water. Phytoplankton account for half of all photosynthetic activity on Earth. Accumulative energy fixation in carbon compounds is the basis for the vast majority of oceanic and also many freshwater food webs. The effects of anthropogenic warming on the global population of phytoplankton is an area of active research. Changes in the vertical stratification of the water column, the rate of temperature-dependent biological reactions, and the atmospheric supply of nutrients are expected to have important effects on future phytoplankton productivity. Additionally, changes in the mortality of phytoplankton due to rates of zooplankton grazing may be significant. As a side note, one of the more remarkable food chains in the ocean, remarkable because of the small number of links, is that of phytoplankton feeding krill, feeding baleen whales. Phytoplankton are also crucially dependent on minerals. These are primarily macronutrients such as nitrate, phosphate or silicic acid, whose availability is governed by the balance between the so-called biological pump and upwelling of deep, nutrient-rich waters. However, across large regions of the world oceans such as the Southern Ocean, phytoplankton are also limited by the lack of the micronutrient iron. This has led to some scientists advocating iron fertilization as a means to counteract the accumulation of human-produced carbon dioxide in the atmosphere. Large-scale experiments have added iron to the oceans to promote phytoplankton growth and draw atmospheric CO2 into the ocean. However, controversy about manipulating the ecosystem and the efficiency of iron fertilization has slowed such experiments. Phytoplankton depend on other substances to survive as well. In particular, vitamin B is crucial. Areas in the ocean have been identified as having a major lack of vitamin B, and correspondingly, phytoplankton. While almost all phytoplankton species are obligate photoautotrophs, there are some that are mixotrophic and other, non-pigmented species that are actually heterotrophic. Of these, the best known are dinoflagellate genera such as Noctiluca and Dinophysis, that obtain organic carbon by ingesting other organisms or detrital material. The term phytoplankton encompasses all photoautotrophic microorganisms in aquatic food webs. Phytoplankton serve as the base of the aquatic food web, providing an essential ecological function for all aquatic life. However, unlike terrestrial communities, where most autotrophs are plants, phytoplankton are a diverse group, incorporating protista eukaryotes and both eubacterial and archaebacterial prokaryotes. There are about 5,000 known species of marine phytoplankton. How such diversity evolved despite scarce resources is unclear. In terms of numbers, the most important groups of phytoplankton include the diatoms, cyanobacteria and dinoflagellates, although many other groups of algae are represented. One group, the coccolithophorids, is responsible for the release of significant amounts of dimethyl sulfide into the atmosphere. DMS is oxidized to form sulfate, which, in areas where ambient aerosol particle concentrations are low, can contribute to the population of cloud condensation nuclei, mostly leading to increased cloud cover and cloud albedo according to the so-called claw hypothesis. Different types of phytoplankton fill different trophic levels within varying ecosystems. In oligotrophic oceanic regions such as the Sargasso Sea of the South Pacific Gyre, phytoplankton is dominated by the small-sized cells, called picoplankton and nanoplankton, mostly composed of cyanobacteria and pico-eukaryotes such as Micromonas. Within more productive ecosystems, dominated by upwelling or high terrestrial inputs, larger dinoflagellates are the more dominant phytoplankton and reflect a larger portion of the biomass. Oxygen production. Phytoplankton absorb energy from the sun and nutrients from the water to produce their own food. In the process of photosynthesis, phytoplankton release molecular oxygen, O2, into the water. It is estimated that between 50% and 85% of the world's oxygen is produced via phytoplankton photosynthesis. The rest is produced via photosynthesis on land by plants. Furthermore, phytoplankton photosynthesis has controlled the atmospheric CO2-O2 balance since the early Precambrian eon. Growth strategy. In the early 20th century, Alfred C. Redfield found the similarity of the phytoplankton's elemental composition to the major dissolved nutrients in the deep ocean. Redfield proposed that the ratio of nitrogen to phosphorus in the ocean was controlled by the phytoplankton's requirements, which subsequently released nitrogen and phosphorus as they are remineralized. This so-called Redfield ratio, in describing stoichiometry of phytoplankton and seawater, has become a fundamental principle to understand the marine ecology, biogeochemistry and phytoplankton evolution. However, Redfield ratio is not a universal value and it may diverge due to the changes in exogenous nutrient delivery and microbial metabolisms in the ocean, such as nitrogen fixation, denitrification and dynamics. The dynamic stoichiometry shown in unicellular algae reflects the capability to stockpile nutrients in internal pool, shift between enzymes with various nutrient requirements and alter osmolyte composition. Different cellular components have their own unique stoichiometry characteristics. For instance, resource acquisition machinery such as proteins and chlorophyll contain high concentration of nitrogen but low in phosphorus. Meanwhile, growth machinery such as ribosomal RNA contains high nitrogen and phosphorus concentration. Based on allocation of resources, phytoplankton is classified into three different growth strategies, namely survivalist, bloomer and generalist. Survivalist phytoplankton has high ratio of N/P and contains numerous resource acquisition machinery to sustain growth under scarce resources. Bloomer phytoplankton has low N/P ratio, contains high proportion of growth machinery and adapted to exponential growth. Generalist phytoplankton has similar N/P to Redfield ratio and contain relatively equal resource acquisition and growth machinery. Environmental controversy. A 2010 study published in Nature reported that marine phytoplankton had declined substantially in the world's oceans over the past century. Phytoplankton concentrations in surface waters were were estimated to have decreased by about 40% since 1950, at a rate of around 1% per year, possibly in response to ocean warming. The study generated debate among scientists and led to several communications and criticisms, also published in Nature. In a 2014 follow-up study, the authors used a larger database of measurements and revised their analysis methods to account for several of the published criticisms, but ultimately reached similar conclusions to the original Nature study. These studies and the need to understand the phytoplankton in the ocean led to the creation of the ʻFaɪtoʊʻplæŋktən citizen science study in 2013. The ʻFaɪtoʊʻplæŋktən study is a global study of phytoplankton conducted by seafarers involving a ʻFaɪtoʊʻplæŋktən and a smartphone. Estimates of oceanic phytoplankton change are highly variable. One global ocean primary productivity study found a net increase in phytoplankton, as judged from measured chlorophyll, when comparing observations in 1998 to 2002 to those conducted during a prior mission in 1979 to 1986. However, using the same database of measurements, other studies concluded that both chlorophyll and primary production had declined over the same time interval. The airborne fraction of CO2 from human emissions, the percentage neither sequestered by photosynthetic life on land and sea nor absorbed in the oceans abiotically, has been almost constant over the past century, and that suggests a moderate upper limit on how much a component of the carbon cycle as large as phytoplankton have declined. In the Northeast Atlantic, where a relatively long chlorophyll data series is available, and the site of the continuous plankton recorder survey, a net increase was found from 1948 to 2002. During 1998 to 2005, global ocean net primary productivity rose in 1998, followed by a decline during the rest of that period, yielding a small net increase. Using 6 climate model simulations, a large multi-university study of ocean ecosystems predicted that a global increase in primary production of 0.7% at the low end to 8.1% at the high end by 2050, although with very large regional differences, including a contraction of the highly productive marginal sea ice biome by 42% in the northern hemisphere and 17% in the southern hemisphere. A more recent multi-model study estimated that primary production would decline by 2 to 20% by 2100 AD. Despite substantial variation in both the magnitude and spatial pattern of change, the majority of published studies predict that phytoplankton biomass and/or primary production will decline over the next century. Researchers at the Woods Hole Oceanographic Institution have found phytoplankton to be a major source of methanol, CH3 oxygen monohydride in the ocean in quantities that could rival or exceed that which is produced on land. Aquaculture. Phytoplankton are a key food item in both aquaculture and mariculture. Both utilize phytoplankton as food for the animals being farmed. In mariculture, the phytoplankton is naturally occurring and is introduced into enclosures with the normal circulation of seawater. In aquaculture, phytoplankton must be obtained and introduced directly. The plankton can either be collected from a body of water or cultured, though the former method is seldom used. Phytoplankton is used as a foodstock for the production of rotifers, which are in turn used to feed other organisms. Phytoplankton is also used to feed many varieties of aquacultured mollusks, including pearl oysters and giant clams. The production of phytoplankton under artificial conditions is itself a form of aquaculture. Phytoplankton is cultured for a variety of purposes, including foodstock for other aquacultured organisms, a nutritional supplement for captive invertebrates in aquaria, culture size sizes range from small-scale laboratory cultures of less than 1 L to several tens of thousands of liters for commercial aquaculture. Regardless of the size of the culture, certain conditions must be provided for efficient growth of plankton. The majority of cultured plankton is marine, and seawater of a specific gravity of 1.010 to 1.026 may be used as a culture air medium. This water must be sterilized, usually by either high temperatures in an autoclave or by exposure to ultraviolet radiation, to prevent biological contamination of the culture. Various fertilizers are added to the culture air medium to facilitate the growth of plankton. A culture air must be aerated or agitated in some way to keep plankton suspended, as well as to provide dissolved carbon dioxide for photosynthesis. In addition to constant aeration, most cultures are manually mixed or stirred on a regular basis. Light must be provided for the growth of phytoplankton. The color temperature of illumination should be approximately 6,500 K, but values from 4,000 K to upwards of 20,000 K have been used successfully. The duration of light exposure should be approximately 16 hours daily. This is the most efficient artificial day length.